Hello students. Today's topic is on virology, the study of viruses. What are viruses? These are unicellular, ultramicroscopic structures which are filtrable. They contain either DNA or RNA and they reproduce inside the living cells. They are metabolically inert outside the host cell. The virus structure is, we should know the nature of the genome, the chemical composition. The size of the viruses varies from 20 nanometers to 300 nanometers. The shape and structure of viruses. The four main morphological forms which are present based on the capsid symmetry are helical, icosahedral, or the complex and bacteriophages. The helical virus, best example is tobacco mosaic virus. Adenovirus is the example for polyhedral virus. Influenza virus spherical in shape, while bacteriophage is complex in shape. The viruses can be classified depending on the presence of the nucleic acid, the RNA or DNA. They can be single-stranded or double-stranded. They can be non-segmented or segmented, linear or circular, and enveloped or non-enveloped. Do all the viruses replicate? And how do the viruses multiply? Few viruses, they remain in the dormant state in the ganglia, that is the latency period. And multiplication of virus occurs by way of first adsorption. They get adhered to the host cell wall. Then penetration of the nucleic acid occurs. Uncoating takes place. And synthesis occurs with the help of mRNA. Assembly and maturation in the host cell, while the release, at the time of release, they take the covering of the host cell. During this stages of virus multiplication, the viruses cannot be demonstrated in the host cell. It is due to the eclipse phase. And formation of these daughter virions, there can be abnormal replicative cycles as well. In some cases, it may give a Magnus phenomenon. What are the effect of viruses on the cells? The host cell can undergo cytopathogenic changes. It can produce virus-specific structures in the host cell. These are the inclusion bodies, which can be intranuclear, intracytoplasmic, or both, as seen in the measles virus. There can be chromosome damage as breakage, fragmentation, rearrangement, or translocation can occur. In some cases, there can be malignant transformation, usually caused by the oncogenic viruses. What is a bacteriophage? This bacteriophage is a virus that attacks and infects the bacteria. Each phage is specific to one form of bacteria. How do phages multiply? They can multiply by the lytic cycle or lysogenic cycle. In lytic cycle, the phage causes lysis and death of the host cell, while in lysogenic cycle, a prophage DNA, it gets incorporated in the host cell. The structure is complex with a head, then tail. It includes the collar and the sheath, while the base plate, it contains spikes or the tail fiber, which gets adhered to the host cell. The various oncogenic viruses are human papillomavirus, which causes cervical cancer. The adenoviridi causes adenocarcinoma. Herpes viridi, in that the Epstein-Barr virus is associated with Burkitt's lymphoma. Human herpes virus A causes Kaposi sarcoma, while hepatitis B causes liver cancer. Now, what are prions? These are not viruses, but they are infectious proteins which are inherited and transmissible by ingestion, transplant, or through surgical instruments. It causes the degenerative changes in the brain, and the disease is mad cow disease or Creutzfeldt-Jakob disease, which includes the disease of dementia and various neurological symptoms are associated. How do we cultivate the virus, or the viruses are grown? Either by animal inoculation, egg inoculation, and tissue culture. In animal inoculation, it can be done via intracranial, intracerebral, subcutaneous. But the problem is it is inconvenient and expensive, not a defined system, and animals can harbor the latent viruses. It leads to generation of escape mutants. Immunity can interfere with viral growth, and not all viruses are grown in animal inoculation. Also, there is animal welfare issues. But the advantage is it gives a unique insight into the viral pathogenesis and host-virus relation. It can be used to study the pathogenesis, the immune response, epidemiology, and oncogenesis. Usually, the various animals used are infant suckling mouse, guinea pigs, monkeys, ferrets, and rabbits can be used. The animals can be made available from Hapkin Institute, Mumbai, Virus Research Center, Pune, Indian Cancer Research Center, Mumbai, and Haryana, that is from the Hisar Agriculture University. For egg inoculation, either the chick embryo or the duck egg can be used. This is the method how the egg inoculation is done. And these are the different parts of the egg. So the choriolentic membrane, pox viruses, herpes simplex virus, and herpes B virus, Rous sarcoma virus can be grown in choriolentic membrane. In the amniotic cavity, the influenza and mumps virus can be grown. Yolk sac, the Japanese encephalitis virus, St. Louis encephalitis, herpes simplex virus can be grown. And allantoic cavity, the Newcastle disease virus or Sendai virus, and again the influenza and mumps can be grown. The disadvantages are, again, only few viruses can be grown in this method. There can be slight contamination which may affect the growth of the viruses or may kill the embryo. But the advantages are it is easily available, cheap, it can be cleaned and made sterile. They do not need feeding and caging. Several sites can be used for inoculation and growth of viruses. The tissue culture, organ culture can be used, explant culture, or various cell cultures like primary cell line, diploid cell line, or tertiary cell line. In organ culture, the tracheal ring is used, as in for coronavirus isolation. For explant culture, the adenoid tissue explant culture for adenovirus isolation or fragment of minced tissue is grown as explants. For cell culture, the primary cell line like human amnion cell culture, monkey kidney or human embryonic kidney cell line is used. In diploid cell strains, the human embryonic lung cell strain can be used. In continuous cell line, it contains cells of single type and serial subcultures are done for unlimited times. In this, the HeLa cell line or HEP2 and KB cell lines can be used. The detection of the virus growth in cell culture can be by the cytopathic effects. There can be crenation of the cells or syncytial formation. Also, focal degeneration or granular clumps can be seen with enterovirus, measles virus, herpes, and adenovirus, respectively. We can use the indicator to find out the metabolic inhibition, which can be detected as the medium turns acidic due to cellular metabolism, and there will be no acid production if the virus is grown as it inhibits the metabolism. The guinea pig erythrocytes adsorb on surface of the cell in heme agglutinating viruses. The various transformation of oncoviruses will be seen piling up, and fluorescent antibody staining or heme agglutination and interference, which the growth can be inhibited by adding a known cytopathogenic virus. These are the ways how to detect the virus growth on the cell culture. The various DNA viruses are either double-stranded DNA viruses or single-stranded DNA viruses. They can be enveloped or non-enveloped. These viruses are the pox virus, which includes the smallpox, or the molluscum contagiosum, in which are cowpox, or virus, Yaba Tanapox virus. Then the herpes simplex virus, which is again a double-stranded DNA virus and is icosahedral with capsid. It is lipid bilayer enveloped virus. There are 8 types: the alpha herpes viridi, gamma herpes viridi, and beta herpes viridi, which includes herpes simplex type 1, type 2, and varicella and herpes zoster in alpha herpes viridi. The human herpes virus 4 and human herpes virus type 8 are included in the gamma herpes viridi, while the human herpes virus 6 and 7 are included in the beta herpes viridi. The herpes simplex virus 1 is associated with the cold sores, oral herpes, herpes labialis, and it is also responsible for herpetic whitlow, keratoconjunctivitis, and herpetic encephalitis. While herpes simplex type 2 is venereal infection produces genital herpes and neonate herpes. The varicella herpes type 3, it expresses the latency associated transcript RNA. The chickenpox is a childhood infection and the latency is seen in the neural ganglia. It resides in the trigeminal ganglia. The chickenpox or shingles can be seen in the later stages. The cytomegalovirus is human herpes virus 5th. Then adenoviruses, they are associated with pharyngitis, tonsillitis, pneumonia, acute respiratory disorders, epidemic keratoconjunctivitis, gastroenteritis, and acute follicular conjunctivitis. The papillomaviridi, they cause warts or cervical cancer. These are the non-enveloped double-stranded DNA viruses. The hepatitis B virus, which is associated with the liver cancer and caused by parental transmission, is enveloped double-stranded DNA virus. The non-enveloped viruses are the parvovirus, which causes the 5th disease. In RNA viruses, these are either single-stranded or double-stranded RNA viruses. Again, they can have positive strand or negative strand. They can be enveloped or non-enveloped. Most of them, they are Papillomaviridi, and they causes encephalitis or respiratory infections. Some are associated with gastroenteritis. So the Papillomaviridi, it includes the enterovirus, rhinovirus, and hepatitis A virus. Calciviridi, it causes the gastroenteritis. Then the togaviridi, flaviviridi, and coronaviridi are associated with the encephalitis. The filoviridi, buniaviridi, and rhabdoviridi are enveloped negative strand single-stranded RNA viruses. Also included are the orthomyxoviridi, paramyxoviridi, and arenaviridi. The rheoviridae family, it causes the gastroenteritis. It is non-enveloped double-stranded RNA virus. These all viruses will be studied in the frequent lectures. Thank you.